Praise the Lord. All right. Yesterday we began our health lessons, and we were looking at the two theories. No. The perspectives, rather, that shape our understanding of disease causation and consequently the intervention we use for disease. And we said what we end up with or what we do is actually based on the evolution theory. And we said it is good as a church to revisit this question of health and try to understand what it is that our message entails. Now, I will not review, our time is really gone, but the Lord will help us. So I'll not review what I did yesterday. I want to get straight into today's lesson. Now, I don't know whether it's reflected on your screens. Is there anything on our screens? Not yet. All right. So I had 2 hours for this program, but graciously I was given an extra hour tomorrow. And I was thinking what to do with 2 hours. So I decided to do what I'm calling confronting the villains of germ theory. Because yesterday I said, if our understanding of disease is based on evolution, then there are microorganisms that we think or we blame for all our diseases. And I said our intervention is usually based on that understanding. So what we usually do is 1, immunize. 2, should you get sick, we have drugs to kill microorganisms. And the third one, we said we try to keep our environment clean. Now, why do we immunize? What are vaccinations for? Yes? Yes, to strengthen our immune system against what? Yes, we say we immunize to strengthen our immune system against viruses. And why do we say we do that? Number 1, because we believe viruses cause disease. That's what we were doing yesterday. And number 2, because we say you cannot medicate against viruses. There are no drugs against viruses. So you need to immunize your body. You need to strengthen your own immune system to fight viral infections. So today, the first villain of germ theory that we are going to confront is viruses. Is it on your screen yet? Yes, it's there. So we are confronting the villains of germ theory. And today and tomorrow, these 2 hours, we're just going to do viruses. Now, let's get into it. Now, when you're talking about viruses, these are examples of what of the diseases that we say are caused by viruses. We have the Spanish flu of 1918, 1919. It was really famous. I've put it there because last year, but one, when we got COVID, we were told that this looks like the Spanish flu of 1918. So we have SARS. It was a thing a few years ago. We have HIV. It was a thing in the 1980s. I don't know whether it still is. We have measles, smallpox, swine flu, avian flu. Now we have that list, and it is by no means comprehensive. There are so many of these so-called viruses. I was just giving examples so we're together. Now, what or who is a virus said to be? Is that the slide on your screens? It is. All right. It's on which screen? This one? This one. All right. Sorry, guys. Pastor is saying you can move that way. All right. I think it would be a good idea so that we can move fast. But if they're going to fix this one fast, it's okay. So those who wish to move can move. Then we can save the the PA team the trouble of fixing this other one. Maybe they can fix it for the next program. Now, my lesson is a bit long, so let me give my apologies in advance. It might take a few extra minutes, but I'll try not to. All right. Okay, now we are told that a virus is an infective agent that typically consists of a nucleic acid molecule in a protein coat. Now, I want you to remember that as we proceed. It's going to be important as we go on. We are told it's too small to be seen by light microscopy. So most of the microscopes that we have cannot help us. We cannot see a virus through that. And we are told viruses either contain genetic material, either DNA or RNA. So it's not a comprehensive description of viruses, but let's move with that for today. Now, how are they said to cause disease? Now, we are told that once they enter the body, they outwit the host cell membrane. In other words, we're told viruses are foreign. They are microorganisms in quotes. They are foreign. So in order to enter your cell, somehow they must trick your cell that they are a friendly amino acid. So we are told they outwit your cell membrane. They pass through the cell interior into the lysosome. Then they trick your ribosomes and polysomes that it's a friendly amino acid, and eventually they multiply. And they kick out a virion or a virion and infect the adjoining cell. Well, basically, that's what we are told. Viruses are, and that's how they infect us. But I've always asked this question. I think for everything that we are told, I think the rational question to ask is, how do we know that? Because if today I come and tell you that I think your neighbor is the one that's causing your headache, he has an evil eye. And the reason you have this headache is because he looked at you a certain way. What's the first question you'll ask me? How do I know that? Did I see him look at you with that evil eye? Can I describe what this evil eye is? So I think it's a question that comes naturally. How do we know that? So if you tell me this happens, then I have a right to ask you, how do you know that that's how it happens? So today we are going to ask ourselves this question. How do we know that? How do we know that viruses exist and that they cause disease the way we are told they cause disease? Now, let's do a brief history of viruses. Now, viruses became a thing in the 1800s. In 1886, we had this German chemist. And at this time, they had a problem with tobacco plants. The tobacco plants were getting a certain disease, and it was affecting the harvest greatly. So they started studying the question of viruses. Now, they suspected that there was something smaller than a bacteria which they could not see. And they wanted to see whether they could isolate another microorganism that was smaller than a bacteria. Because at the time, they could isolate bacteria. They could see them, they could classify them, they could study them. But they suspected there must be something else other than bacteria that's causing this disease. So they started doing it with tobacco plants. In 1892, that study was improved, and in 1898, the term virus now was invented. Now, what they called it was Contagium vivum fluidum, simply meaning a poisonous fluid. So virus simply means a poisonous fluid. It's not a reference to any living thing. Now, the trouble with that initial experiment, I'm not going into detail. Anybody who is interested maybe can see me and I can share the material. Now, what they did, they filtered. They took the leaves of diseased tobacco plants. They filtered them to get rid of bacteria and other larger microorganisms. And then they went and tore the leaves of healthy tobacco plants and applied that fluid. And some of the tobacco plants died. And from that experiment, they said, now see, we have something in this fluid that is causing the tobacco plants to die. Now, the problem with that is, number one, they did not isolate any living microorganism. So there was nothing they could say, this is the reason the tobacco plants are dying. Number two, if you damage the leaves of a healthy plant, even without applying anything, what happens? It dies. So there was no control experiment to say, we damaged the leaves of this plant and did not apply this which we suspect is causing disease. And then we damaged the leaves of this one and applied what we suspected is causing disease. And this one survived and this one did not. There was no control experiment. But they still went ahead and said, we know there is something in this fluid. There is something here that's causing the plants to die. Now, when challenges were raised at the time, and they were asked, questions were raised about whether that experiment would pass scientific standards, now some issues were raised in return. And number one, they said viruses are so small, they cannot be filtered. So there was no way they could isolate a virus then. And we are told that viruses are so small, they are measured in nanometers. Now, you can see how big a nanometer is on your screen. Now, 1 nanometer is equal to 0.000000039 inches. Now, how small is that? How small is that? Is it something you can... Now, let's bring it home. For example, we're told the poliovirus is 30 nanometers across. Now, that's 10,000 times smaller than a grain of salt. Now, I want you to take fine salt, isolate 1 grain, divide that into 10,000 pieces, and you're told you will have the size of a polio virus. So those were issues then, during those first experiments. Then in 1931, we got the first electronic microscope. In 1939, it was mass-produced. Sorry, it was mass-produced in 1931. Then in 1939, we were told that now, because of the introduction of electronic microscopy, we could, for the first time, get clear snapshots of an altered virus. So we could see a virus clearly and in a form that was not altered. Those were the claims in 1939. Now, it is good to see what exactly that statement means. Because when we say electron microscopy shows us clear and altered images of viruses, then it would be such a privilege for us to look at them. Isn't that so? All right. Now, I want to look at the screen. Now, that's an image of the rotavirus. Now, what does the rotavirus do? Or what does it supposedly do? It causes diarrhea, we are told, especially among little children. So for most cases of diarrhea, you're told your child has what? Rotavirus. Now, that first image, we have A, B, C, so we have different magnifications of that same image. So in A, we have that kind of magnification. Now you can see the red circle somewhere. Yes, can you see that virus clearly? Now, I hope God's people are honest. Can you see it clearly? You're seeing nothing. All right. Now, that is a computer-generated image of rotavirus. Now, this does not exist in nature. This is created by a computer. Now, every time we are taught about viruses, this is what we are shown. But this does not exist. It only exists in the mind of the person who does what? Creates it. It's created in silico. It is computer generated. Now, this is what we are told is SARS-CoV-2, or what we come to know it as COVID-19. Sorry, no, not COVID-19. This is COVID-2. Now, this is the electron microscope image. What do you see there? Now, those white blots you're seeing there are not the viruses. Besides them, there are nanoparticles that are supposed to be the viruses. Can you see them clearly? Now, if you tell me you cannot see them clearly, I will tell you you do not have a scientific mind. Now, can you see them clearly? Now, all those who want to have a scientific mind will tell me yes. You cannot see anything. You can see blots. All right. Now, that's the computer-generated image. And every time they want to tell us about the virus, this is what? That's what we see. Now, recently, I think it was this year, we were told there is another outbreak of Marburg. You heard about that in Tanzania? We were told there is a new outbreak of viruses. So there is a virus in Tanzania called Marburg. Now, every time you look at it from the electron microscope, that's what you see. That thing that looks like a bone that a dog ate and left. That's what they see. Now, this is what is used to educate the public about the Marburg virus. So when we say there are clear, the electron microscope gives us clear images of viruses, that is what we mean. That's what's meant by that. Now let's look at the second issue with the electron microscope. Not the second issue, on that issue. Now we are told viruses have no color and that they are smaller than a wavelength of light, which means they are so small, the only thing that can be seen are undefined images of nanoparticles. Nanoparticles simply means really small particles. That all we see are small particles on that image. Now, the second problem with electron microscope, with electron microscopy is that in order for you to prepare an image to be examined, you must have some chemical preparation and examination. And this is one way in which you prepare that sample for examination. So the samples are negatively stained by adsorbing them in a fluid on a plastic-coated grid. Now, you can read the rest of the process. I don't want this lecture to be too tedious. So you have a chemical process that preparation must go through. And this is one way. The other way is the one on your screen. So the specimens, instead of that initial chemical preparation, are fixed in formalin, they are stained, then they are embedded, they are baked to harden, then they are sliced really small, and then eventually now you can put them under your electron microscope. Now, the trouble is, once you do that, the chemical preparation itself alters whatever specimen you had, such that at the end of the process, you cannot claim that what you end up seeing is anything related to what you thought you were looking for. Now, the second problem is the amount of radiation received by a specimen during the EM imaging is similar, we are told, to the irradiation from a 10 megaton hydrogen bomb exploded 30 meters away. Now, if we had that exploded 30 meters away from here, what would happen to us? Would there be anything left here to examine? And yet, that's what's done to all specimens. And then at the end of that process, we are told that the result we see under that electron microscope are what? Viruses. Now, I think any thinking person will admit that electron microscopy is not a diagnostic tool. We cannot tell that viruses exist simply because we have electron microscopes. But electron microscopes do show that there are nanoparticles. There are small grainy things on that slide. So how do we know that those small particles are viruses? How are we supposed to know that? The truth is, we do not know. For example, let me show you these two images here. Now, this is an image of what's supposed to be COVID-19. You see where the arrows are pointing? Now, this is highly magnified. So you see those dark circles there? Now, let me show you this. You see the same dark circles here? Now, this is damaged kidney tissue. This, we are told, is COVID-19. Is there any difference between those grainy images? There is no difference. And in fact, if you delve into scientific literature, you will find so many concerns of people who are diagnosed with COVID-19 when all they had was damaged kidney. And they could not tell the difference, because you cannot tell the difference using electronic microscopy. So we do not know. And the truth is that the products of electron microscopes are at best unclear and highly altered. So that does not tell us about the existence of viruses. So let's go to the next tool. How else are we told we know that viruses exist? The next way they tell us they can tell without doubt that there are viruses and they are causing all the problems we are seeing is this instrument called what? The PCR. Now, what's the PCR? PCR simply stands for polymerase chain reaction. You can study more about it if you're interested. Now, the truth is, the PCR is very sensitive. It's a very sensitive tool. And as long as there is genetic material, the PCR will find it, and it will tell you that there is genetic material. The problem is, it does not tell you where that genetic material comes from. So all it tells is that whatever you've given me, there is genetic material. But the origin of that genetic material cannot be told using the PCR. So the only way the PCR can tell us that we have a virus is if first we isolate that virus, we profile it, we get to know what genetic sequence it has, then when we put it in the PCR, we can tell that this genetic sequence from this virus is in this sample that you have given us. So the PCR is a good thing, but it does not tell us anything other than the fact that there is genetic material. That's all it tells us. So we must first have isolation and purification of the so-called viruses. Now, let's talk about that a little. What do we mean by isolation and purification? Now, it is as simple as it sounds. When we talk about isolation, we are talking about separating something from other things. Now, if I want to isolate you, I don't even need to do that. There are 2 people in this congregation who are already isolated. You're welcome to join the others. But if I wanted to isolate you from this crowd, what would I do? I would ask you to stand, then I would ask you to sit separately. It is that simple. Now, what is purification? Now, once I have isolated something, I need to make sure it is not contaminated by whatever was surrounding it in the place where it was. Now, I like using this example. What do you see on your screens? I know it's far from lunchtime, so nobody is hungry. I am not torturing anyone. But what does that look like? I think it will look like different things to different people. But what does it look like to you? Githeri. Githeri and avocado. And my githeri has a lot of mixes. Huh? It has carrots. I don't know what that other thing is. Looks like potatoes. So now I have this mix here. Now suppose I eat this for lunch. And then 2 hours after eating it, I start making these frequent visits to that office down there. And then you ask me what's wrong with me, and I tell you, I am sure the maize in that githeri was the problem. Now, what will be your question? How do I know it was the maize? What should be the right answer? I should tell you, I have isolated what? The maize. Or what should be the process, rather? Now, if it's the maize I am suspecting, what should I do to it? I should take that same githeri that I ate. I should isolate the maize. I should pick the maize from the githeri. After picking the maize, that's isolation, right? I should wash it so that the bean soup and the carrot soup and the stains from the avocado and whatever else was in that mix is no longer in the maize. So I'll do my best to make sure that the only thing I remain with is clean maize. I am purifying my sample. And then I should eat this maize and wait 2 hours and see whether I still make what? Those frequent trips to that small office there. Now, if that does not happen, what should I do? Suppose I say, now I know it's not the maize, it's the beans. What should I do? I should come back, take that same bowl of githeri, and pick out the beans. Now, if those don't affect me, and I come back and I say, I know for sure it is the carrots, then I should repeat that same process until I isolate, purify, and test that which I suspect to be the problem. Now, that process is scientific, and the Lord has no problem with science. In fact, science is biblical. Now, read this statement with me. This is Counsels to Parents and Teachers, page 295 to 300. You can read it in your own leisure. It says, the Lord bids us reason from cause to effect. What's that? That is a scientific process. It says, reason from cause to what? Effect. Establish the cause of the problem. Now, to remember that we are his property and to unite with him in keeping the body pure and healthy and the whole being sanctified to him. So we have no problem with the scientific process. It is God who bids us be scientific, establish the cause of disease. Now, in 1890, while science was still sane and a little biblical, not all of it was. Now, while it was still sane, they came up with this, 1890. They called them Koch's postulates or principles that should be followed in establishing whether something, actually specifically microorganisms, cause disease. They said if you want to blame any microorganism for causing disease, you must establish, you must meet these 4 principles. That was 1890. Now, for so long, they tried all the way until 1937. They tried to establish disease causation. They failed to establish that microorganisms were causing disease. So in 1937, they actually revised the postulates and said, okay, the 4 postulates as they stand cannot serve us anymore. Now, if you are a Christian, maybe you'd probably think, maybe we are not meeting the standards here of these 4 postulates simply because microorganisms don't cause disease. But no, they said, no, it's not because microorganisms don't cause disease, but the manner in which they cause disease must be more complicated than we know. But for today, I want us to use those 4 postulates. Now, number 1, is it still on your screen? Yes. So number 1, it says the microorganism must be detectable in all cases of disease with the same symptoms, but not in healthy individuals. Now, let's say there are 4 of us here who are coughing. And we say, we suspect these people have tuberculosis. They have TB. And we are sure that what's causing the cough is the TB germ. How should we be able to establish that according to the first Koch's postulate? Now, in all 4 individuals who are coughing and have the TB germ, okay, we should detect the TB germ in all those cases that are coughing. That's the first point. Then the second part of that postulate is that when we go to the healthy individuals, we should not find that germ. Fair enough, don't you agree? Yes. So if I think the cough is being caused by this particular germ, it should only be present in those who are coughing, but not those who are not coughing. I think that's fair enough. The second postulate, I should be able to get that TB germ, isolate it from the sick person and put it in someone who is not sick. And when I do that, the third postulate states that that previously healthy individual, if given the same germ that was causing the cough, or I was suspecting was causing the cough in this other one, they should also start doing what? Coughing. Is that fair so far? Is it rational? Is it scientific? All right. And then number 4, the fourth postulate says, that same microorganism, now in this new individual whom I have infected, I should be able to transfer it from him to back to a pure culture, and I should be able to replicate that process of infection throughout. Now, when it comes to these viruses, one, we said there must be a process of isolation and purification. Then once you do that, you should infect people using those 4 postulates. Now let us look at this history. I know my time is really gone, otherwise Pastor wouldn't be here. But let's forget time for just a little while. Now let's look at the history and the history of purification and isolation of viruses. Now this one was the measles virus. And this happened in 1954. We had these 2 guys. First one was called John Enders, the other one was called Thomas Peebles. And they put enormous effort in trying to grow viruses. So they tried doing it on the human lung. They couldn't. They tried intestinal cells. They couldn't. They tried cells from the uterus. They failed. They tried cells from the skin, from the muscle, even from the foreskin. But no viruses could grow. They tried embryonic chicken cells. They tried monkey cells with no success. Finally, they said they had succeeded by using human kidney cells and cells from the rhesus monkey. And they said the reason they knew they had succeeded is because they achieved what's called cytopathic effects in the cells they were using. Now, cytopathic effects, it's just a big word to say there was cell damage, that the cells they were using were damaged, and they were sure the damage was caused by what? By viruses. Now, let us look at that process. Now, there are people who are, especially the young people here, who are very curious about how to make a virus and annihilate the human race. Now, here is a formula on how to make a virus. Follow closely, and you might just make your own virus. Now, number 1, the first step. Now, we will all be scientists today. Now, number 1, they took swabs. Simply means just take a swab of cotton, and then you take samples from the throat. Now, most of the time when you have measles, you usually have a sore throat. And it's suspected most of the viruses will be lodged in your throat. So they would take swabs from your throat, and then they would take blood samples. Now, are you still with me? Now, where I lose you is where I'll stop. Are you still with me? All right. So we are taking our sample now. So we are taking our sample from the throat and from blood. That's our sample. Now, this, which we have taken, was immersed in cow milk. Now, why cow milk? For nutrition, to keep the cells alive. Then they were given a dose of penicillin and streptomycin. After that, they added a little bovine amniotic fluid, beef embryo extract, and horse serum for additional nutrition to keep the cells alive after poisoning them with antibiotics. Then after that, they added more unspecified antibiotics. Then they used phenol red to stain that. They used formalin to preserve it. They mixed ethanol and ether. Then they stained it a little further. Then they washed that same sample in 95% alcohol and oil and topped it with xylene. Xylene is a chemical that's supposed to eat away the tissue so that you end up with what you want to see. All right? Now, after that comprehensive process, we are told cytopathic effects were done what? Were observed. What do we mean by that? After that comprehensive process, the cells were damaged. And because the cells were damaged, it became proof that the measles virus had done it. Now, if you were the measles virus, what would you say in your defense? If I were a virus and I was suspected of causing that disease, what would I say in my defense? Number one, I would tell you that the penicillin and the streptomycin that you put on your cells was enough, even without adding formalin and the 95% alcohol and all the other soup of toxins that you added to the cells. Even without that, just the antibiotics alone were sufficient to cause cytopathic effects. If I were in court and I was a virus, would I go free? I would go free without any other evidence. So in our history of isolation and purification, there are plenty of lies. Now, there are obvious challenges, as we have pointed out. There was no isolate. Now, let me take you back to this page. Now, if I were to take a sample of this and put it in the PCR, would I get genetic material? Just think with me. Would I detect genetic material? Everyone is quiet. Would I detect genetic material? Of course, there is plenty of genetic material. The first would be from the person, the supposed patient, from the throat and from the blood. And there would be genetic material from the cow milk. There would be genetic material from the amniotic fluid. There would be genetic material from the beef embryo and from the horse serum. I have a soup of genetic components. So if I put that in the PCR and tell you I have found genetic material, RNA in nature, so I am sure I have found the virus that's causing you problems, what would you tell me in all truth? One, you would begin by disqualifying me from being a scientist. You would say I did not follow due process in establishing that fact. Now, let's continue. So there were obvious challenges. And because of those challenges, they asked this professor, what exactly do you mean when you say you have an isolate and you can tell us about this SARS virus? And this was his statement. I find it quite interesting. It says an isolate is a virus that we have isolated from an infected host, and we have propagated that in culture. Now, when I say we've propagated that in culture, I'm talking about that process that I've just taken you through in establishing or in finding a measles virus. That genetic soup there that has been poisoned with antibiotics and preservatives and alcohol. So that's what they propagate in culture. Now you put that sample in cells and culture. And that's how you grow viruses. Now, in the previous two slides, did we grow viruses? Did we grow anything? All right. Now, that's how you grow viruses, and that's how you have a what? An isolate. Now, I know I am talking to men who think. Now, does that statement tell you anything? Don't tell me what it tells you. But we do know that in that process of growing viruses, the cytopathic effects, the damage to the cells that we are told are caused by viruses, can be caused by almost all the ingredients that are used in that cultures. In fact, in 1954, they admitted that the cytopathic effect, which superficially resemble those resulting from infection by the measles agents, may possibly be induced by unknown factors. Now, what kind of statement is that? What would have been the honest statement there? It would have been, we did not find a what? A virus, but we did manage to damage cells using antibiotics. That would have been far much more honest. But okay, honesty is not the name of the game. 2015, we had another controversy. Now, Dr. Stefan Lanka is a German scientist. He is actually a virologist. And for the longest time, he thought he was studying and seeing viruses. Then in 2015, the German government mandated the measles vaccine for all children. And he started studying it. Now, he was not studying it because he didn't believe viruses exist. He was studying it because he did not think it should be mandated. And he wanted to advise the government and tell them, I don't think measles is that dangerous, so let's not mandate it. Let's just give to those who really need it. But in the process of studying it, he actually managed to achieve those same cytopathic effects without a sample. And then he realized, actually, that what we use as proof that viruses exist is actually not proof. There is no proof in it. Now, he actually offered 100,000 euros. Now, how much is that in Kenyan shillings? You calculate it. I need to calculate it. Now, I know, how much time do I have left? I don't want to disrupt the whole day's program. It's over. Okay, I have no more time. So it's okay. I think I'm going to stop here. But the point I have in mind, I will finish tomorrow. We will make no conclusions today. But tomorrow, and tomorrow's program starts a little earlier. It's going to be 8:40 to 9:40. We are going to finish this thought and finish our confrontation with the first villain of germ theory. May God bless you.